In the case of photosynthesis, remember that it is being accomplished in two major steps. The first one, the presence of photo, and the second one, the presence of synthesis. And when it comes to this case, we are going to focus first on the keyword photo, which is also known as the light reaction. Now, this is actually the phase 1 of photosynthesis. The phase 1 of photosynthesis, it is also known as the light reaction or the light-dependent reaction. And it is happening on the thylakoids that is present among chloroplast. Then, what are the different things that will simply happen here? On phase 1, the split of water due to the process known as photolysis will happen, the release of oxygen gas, the reduction of NADP+ to charge nicotinamide adenine dinucleotide phosphate and generate adenosine triphosphate from adenosine diphosphate by the process known as photophosphorylation. So these are actually the different things that we are going to consider for phase 1. In the case of the presence of the light reaction, majority of the light capturing moment, it is actually being accomplished on the membrane that is present on the thylakoid. And remember, on the membrane, it is known as the thylakoid, while the interior of the thylakoid is now known as the lumen, in which I am going to discuss later on. Okay? Remember, in the case of the thylakoid, there is now the presence of this complex protein known as photosystem. And the photosystem, it is now containing the chlorophyll. Okay, so if the chloroplast is containing thylakoid membranes, in the thylakoid membranes, there are proteins embedded known as the photosystems. Inside these photosystems, that is the only place where you can locate the chlorophyll. Okay? The chlorophyll pigment is present on the photosystems. There are actually two types of photosystems. There is the presence of photosystem 2 and photosystem 1. The reason why photosystem 2 and photosystem 1 is named that manner is because photosystem 1 is actually discovered first before photosystem 2. So that's basically the reason. And in the case of their names, photosystem 2, it is known as the plastoquinone oxidoreductase, while in the case of photosystem 1, it is known as the plastocyanine ferredoxine oxidoreductase. So when it comes to their different names, they're actually related to different enzymes that is present near and adjacent to every single one of them. But I won't actually complicate things since it is already present on your modules, and I'm actually going to discuss them later on in this video. So remember, in the case of the photosystem, it is responsible now for the harvesting of the energy that is present on the light in the form of photons. So we are considering here that light is a particle in the form of photons. In the case of the particles of light known as photons, it will actually excite now the different types of chlorophyll that is present and embedded in this photosystem. On the part known as the light harvesting complexes, there are different types of chlorophyll that are already scattered. And the function of this chlorophyll is to transfer the energy that's coming from this photon one chlorophyll to the next until it reaches the reaction center complex. The reaction center complex or the reaction center chlorophyll, it is now a special pair of chlorophyll A molecules that will energize the electron that is present in this photosystem. Okay, this is happening both on photosystem 1 and on photosystem 2, but their differences, I'm going to explain this later on. Okay, in the case of the photosystem, they are always responsible for energizing electrons. Always remember that. Do not forget. They're responsible for energizing electrons, both photosystem 2 and photosystem 1. In the case of the photosystems, they will always energize the electrons that is present on the reaction center complex. Now the question is, where is the source of those electrons? Those electrons were simply sourced by the process known as photolysis. And photolysis will actually happen on the lower portion of the photosystem. Okay? And in the process known as photolysis, it will now split up water molecule, and oxygen will be placed on the atmosphere, while the hydrogen ions will remain on the lumen, while the extra electron will now be utilized on the reaction center complex. For familiarity of the different proteins, once again, we have here the presence of the thylakoid membrane. Okay, you can observe here the thylakoid membrane. The interior of the thylakoid is known as the lumen, and the space and the fluid that is filling in the chloroplast, it is now known as the stroma. Okay, stroma exterior, thylakoid the membrane, lumen inside the thylakoid. Now, when it comes to the different proteins that are present on every single one of them, the first one, we have here the presence of the PS2, or also known as the plastoquinone oxidoreductase. Then, we have here the presence of a mobile electron carrier known as the plastoquinone. Then the next one, we have here the cytochrome complex. Then the third one, we have here the presence of the plastocyanine. Then the next one, we have here the presence of PS1 or photosystem 1, or also known as the plastocyanine ferredoxine oxidoreductase. Then the next one, we have here the presence of the ferredoxine. Then we have here the NADP plus reductase. And lastly, we have here the presence of the ATP synthase. So these are actually the different proteins that are responsible for the process of the light-dependent reaction. Now, what will simply happen? The very first thing that will happen, we are going to focus here. For the first step, the sun will bombard by its photons the reaction center of the photosystem 2. Okay? And the photons will now excite the chlorophyll. The energy will transfer one chlorophyll to the next until it reaches the reaction center. While that is happening, the water that is present on the lumen will move towards photosystem 2, and the process known as photolysis will happen, in which the split of water molecules to hydrogen ions that will stay on the lumen, the electron that will stay on the photosystem 2, and the oxygen that will be released on the atmosphere. Okay? But once again, we are just going to focus on the electrons that is present on the photosystem 2. The electron will now be energized because of the reaction center chlorophyll. And since it is highly energized, it will now be transferred by the plastoquinone that is beside the PS2 to be transferred to the cytochrome complex. Okay, now for the second step, the highly energized electron that is now present here on the plastoquinone will be transferred on the cytochrome complex. Remember that majority of the hydrogen ions are already present in the lumen. So the lumen is now possessing high concentration of hydrogen ions. But because of the energy that is present on the electron, it will actually activate the cytochrome complex for the hydrogen ions present on the stroma to move inside. Okay, please take note. The hydrogen ions will now move from an area of low concentration, which is the stroma, to one area of high concentration, which is the lumen. They will do active transport. So, if you can still recall in our discussion on module number 5, this is actually one of its applications. On module number 5, we discussed here that in the case of active transport, they are going to move from an area of low concentration to an area of high concentration. And this is actually an application of the active transport. So now, the energy that is present on the electron, just follow the cursor, it will now be low since it is now already utilized by the cytochrome complex. The next electron carrier, known as the plastocyanine, will now move the electron, the low energy electron, towards the photosystem 1. And since this is a photosystem, you know what will happen. It will now activate the electron that is present, and it will now be highly energized. the highly energized electron will now move on the ferredoxine, and the ferredoxine will now transfer it to the NADP plus reductase. So that is the end of step number 3. Now, on step number 4, since this is now the NADP plus reductase, the NADP plus that is lodging or present here will now attach itself to the highly energized electron. And this highly energized electron will now create the charge NADP plus and will now create the NADPH, or also known as the charge or reduced nicotinamide adenine dinucleotide phosphate. And the electron will now be gone on the thylakoid membrane. Okay, so the electron will be passed from photosystem 2 up to the NADP plus reductase until it reached the final electron acceptor known as the NADP plus to create NADPH. Now, on the last step, since hydrogen ions moved in from the stroma to the lumen and there is now very high concentration of hydrogen ions here, hydrogen ions will have no choice but to move towards the ATP synthase. ATP synthase is actually just like a gear. Once the hydrogen ions move towards this protein, the free phosphate and the adenosine diphosphate that is present on the stroma or that is lodging here near the adenosine diphosphate will now be attached with each other, creating now adenosine triphosphate. So basically, the process is just like this. In summary, photolysis, then the highly energized electron will now be moved by the plastoquinone to the cytochrome complex. The hydrogen ions will now move from the stroma to the lumen. Then more high concentration of hydrogen ions will happen here. Then the low energy electron will now be transferred by the plastocyanine to the photosystem 1 to be re-energized. Then it will be transferred by the ferredoxine. Then on the NADP plus reductase, it will now be accepted by the NADP plus to create NADPH. While the hydrogen ions that entered from the stroma to the lumen will now be released on the ATP synthase, creating now adenosine triphosphate from adenosine diphosphate and the free phosphate that is present on the stroma, creating now ATP. And both of which will be utilized on the next step known as the Calvin cycle. And at the end of our consultation period, I'm going to provide a summary of the different things that is actually present when it comes to the concept of the light-dependent reaction and even of the Calvin cycle or the light-independent reaction.